All right, everyone, thanks for coming. I've got the pleasure of introducing Dr. Zach Schlader. Just a little background, he's an associate professor in the Department of Kinesiology at Indiana University in Bloomington. Comes from an exercise science background like many of us. He completed his PhD at Massey University in New Zealand, where he first kind of got his teeth into some thermal regulation, talking about or investigating the effects of temperature and behavior of temperature on thermal regulation. But he's originally from Columbia, Missouri. After his PhD, he completed his postdoc training with Dr. Craig Crandall, UT Southwestern, so big thermal regulation environmental physiology lab. So he's had a very narrow focus in terms of his interests as such are environmental physiology, with particular emphasis on thermal regulation and just general health and physiological function. He's well funded through various organizations like the CDC, got some DoD grants, as well as National Science Foundation. Several publications, got well over 100 publications. Many honors, just to name a few. He's a fellow of the American College of Sports Medicine. He's on the editorial board for AJP Reg. He's also the thermal counselor for EE through APS. That's pretty interesting. He's also a Division I college basketball player. So Zach will be, Dr. Schlader will be presenting his research entitled "Too Hot to Handle." Very provocative title: heat stress, heat waves, and the kidneys. So thanks, everyone.
Thank you very much for that introduction, and I'm so excited to be here. So thank you, everyone who was involved in getting me here. It's really cool to come back to Columbia. I haven't been back in a while, and so it's cool to see it a little bit from this angle. So I'd like to start with, I had this title, "Too Hot to Handle." I thought, oh, that's kind of clever. Subsequently, like I've talked since the spring, and I'm kind of using little tweaks to this. And so as I was putting this together, I was like, oh, maybe I should have like a picture of some sort. So I Googled "Too Hot to Handle." And apparently there's a Netflix series, reality series on this. So there are no really good images unless they're clearly. Yeah, maybe that would be good. Yeah. So maybe I'll do that next time. Okay, so let's go ahead and get started. Before we really get going, I want to, that way they don't get lost in the shuffle. I would like to highlight the three trainees that have been really instrumental in this. None of this work could be done without them. The first is Chris Chapman. He was a PhD student of mine when I was at the University at Buffalo. So for the first 5 years of my career, I was there. And the last 3 years, I've been at IU. Subsequently, Hayden Hess is a current postdoc in the lab at IU who's just went on a job interview yesterday. So we'll see how that went. And then Jess Freemus finished her PhD over the summer. So you'll see their names around this. I just wanted to make sure they get highlighted early and often. So this is what we're going to talk about today. We're going to talk about acute kidney injury as a public health problem in the context of heat. We're going to talk about how we study that in humans, because our lab at this point is mostly human. We do have some collaborators doing some work in rats, but that work's not quite ready for the prime time yet. And then we're going to talk about, most of our work is the mechanisms by which heat stress may elevate acute kidney injury risk, and then we'll summarize. So let's start with the top. So hopefully figures like this don't surprise people anymore. It's getting hotter. So this is the average global temperature. And this is a departure from let's say the early 1900s. And you can see it's clearly getting hotter on a global scale. In fact, the last 10 years have all been the 10 hottest on record. 2021 sat right in the middle of that. So clearly temperatures are going up. Global temperatures are going up. This is average surface temperature. And the most recent IPCC report said, look, even if we get carbon emissions under control, this is still going to go up over the next 50 to 100 years. So we're not going to meet our benchmarks, which is obviously unfortunate. There's a number of different factors, different things that this global warming brings about. Probably the most overt thing is we have an increased frequency, intensity, and duration of heat waves. So that's what this shows here. This is some data from NOAA showing heat wave frequency is increasing over the decades. So from the 1960s to the 2010s, Heat wave duration is getting longer. The heat wave season, so heat waves are happening earlier. We start to see extreme heat in May and June. For instance, last summer, the extreme heat wave that hit the Pacific Northwest hit in June when they're not ready for those types of temperatures. And also the intensity, so how hot it's getting during these is going up. And this is probably the most striking evidence that I can find as far as in a figure. So what does this mean for health? I could pick any number of figures. I think this one is one of the most pretty, so we put this one up. But here we have the relative risk of all-cause mortality, and on the x-axis is ambient temperature. And hopefully what you can appreciate is that above a temperature of about 25 degrees Celsius, you see a marked increase in the relative risk of mortality. This same figure could be used for morbidity, it could be used for hospitalizations, you name it. And it's really across organ systems as well. Most of the deaths are occurring due to cardiovascular events. A lot of the morbidity and mortality morbidity is happening on the dehydration and kidney side of things, which is what we're going to focus on today. So to transition a little bit to our work, it's really initially founded on this epidemic of chronic kidney disease that's occurring. It was first identified in Central America, in the sugarcane fields in Central America. After it was identified, you can see hot spots have been identified throughout the globe. Essentially, follow the equator, intense manual labor, largely in agricultural settings, because that's where the intense manual labor sits. And you can find these hotspots of CKD. Now, these people are young. They're all usually younger than 45. They are not obese. They're normotensive, and they do not have type 2 diabetes, all of which are the top risk factors for CKD. So epidemiologists have tried to first identify what could be going on. So the first thing they do is that they've looked at agrochemicals, pesticides. Surely maybe there's something they're putting on the crops that is hurting these people. Doesn't look like that's the case. Now, there's maybe an argument, a slight argument for interactions between different things. But generally, if you look at the levels of these pesticides that are showing up in people's urine, they're way lower than what you might expect with other epidemics of chronic kidney disease. Other things like water contamination. Sure, the water is probably not as clean as it is here, but the levels that they see of certain metals do not rise to the extent that would typically be seen with CKD. But this has led to the top hypothesis is that repeated heat stress and dehydration can cause chronic kidney disease. And there's a number of different epidemiological studies that have highlighted this, but probably the most striking evidence was a preclinical study. So here, some data from Roncal Jimenez from 2014, which now is a long time ago, but this is still really innovative. And to my knowledge, this is the first study to show that heat and dehydration can cause CKD. So what they did is they exposed mice to 7 hours per day of 5 days per week for 5 weeks to heat. And in one group, they allowed them to drink water anytime, so they could drink throughout that heat stress. In the other group, they could only have water 1 hour after that ended. So essentially overnight, they got as much water as they would like. And then they had a control group. So they weren't exposed to heat and they were allowed water all the time. So what did they find? Well, first, this is just one of the markers that would be indicative of chronic kidney disease. This is renal interstitial fibrosis. And hopefully what you can appreciate here is that this amount of interstitial fibrosis is much higher when these mice were not allowed to drink during the heats. But if they were allowed to drink during the heats, this largely normalized this response. So there was no statistical difference between the control group and the water anytime group. So it does appear that repeated bouts of heat stress and dehydration can bring about CKD. If you further look into it, you can see here we have, this is the loss of tubular brush borders. So this is indicative of renal tubular injury. And what hopefully you can appreciate is in the water after group, they have much lower levels of this tubular brush border. So this is a marker really of acute injury. And this really led to this hypothesis that heat stress and dehydration increases the risk of kidney injury. So it provokes some amount of kidney injury. It's likely that this is not an amount of kidney injury that you would be admitted to the hospital with. This is something subclinical, whatever that means, right? There's a lot of disagreement in the literature of this. And when this AKI is repeated over time, we're talking weeks, months, years, that it can manifest as chronic kidney disease. And this is really the first time that heat stress and dehydration have been linked to acute kidney injury. So from this evidence, a number of different studies have come out really over the last 2 to 3 years, which have tried to understand what's happening in workers or what's happening in the general public with regards to heat exposure and dehydration. And this is just one of them. This was published, I think, in late last year, where I would like to highlight that these are workers in the United States and that workers-related heat-related kidney injury in a variety of industries, is both indoor and outdoors, when they're exposed to heat. So the hospitalizations for kidney injury are present in the United States. We also have data over here, which are really, think of this more as heat wave data. They're just equating the ambient temperature to hospitalizations for acute kidney injury. And lo and behold, we see as ambient temperature goes up, we see a rise in the risk of developing AKI. So it's really this epidemic, or my connecting of the dots is this epidemic really then highlighted, maybe we need to be looking at whether there's something going on here with heat and dehydration and what's happening with the kidneys. Okay, so now we're going to transition, spend some time on how we study AKI risk in humans. Notice I'm not actually saying AKI because I like to think and I hope that we're not actually giving people acute kidney injury. But before we get into that, we need to talk a little bit about the physiology or the renal physiology of heat stress, dehydration, and or exercise, particularly when we're talking about workers. So here we have the kidney, the renal pyramid. If you zoom in on that, glomerulus, we have our proximal tubule, distal tubule, and collecting duct. So when our body temperature is neutral, much like it is now, our body water's steady, or even if we drank a bunch of water and we're just resting, this is what the renal physiology looks like. So our kidney function, namely glomerular filtration rate, hasn't changed. It's neutral. In a healthy person, you're talking about 100 or more milliliters per minute. We have relatively high urine production, and our urine is not very concentrated, so it's pretty clear. In this case, our kidneys receive a lot of blood flow. So it receives about 25% of the resting cardiac output. Now, what's interesting, however, is that the kidneys do not receive that blood flow homogenously. So the renal medulla is actually has relatively low blood flow, whereas the renal cortex has a relatively high blood flow. And as a result of all of these things, the fact that we're well hydrated and not exercising, we have relatively low levels of vasopressin or antidiuretic hormone or ADH. The RAS system is largely turned off, and there's low levels of renal sympathetic nerve activity. And all of these things contribute to sodium reabsorption. So think of it as this: our body reabsorbs sodium, and the water follows. So it helps us preserve body water. And in this case, we're not really absorbing or reabsorbing very much sodium. So this is what allows us to have relatively high urine production and a relatively low urine concentration. So I'll switch to instances of heat stress, but you could also layer on reductions in body water or dehydration or even exercise. And these things tend to happen. We'll discuss a little bit about what the relative contributions of these things are. But in this case, you see reductions in GFR. You see reductions in urine production and increases in urine concentration. And these are good things. When we're hot, we're sweating, we're losing body water. And so our body says, hey, we need to hold on to as much of that as possible, right? If we're dehydrated alone, again, our body has that same response. We need to hold on to fluid as well as during exercise. So next, I want to kind of highlight this relative role of body temperature and dehydration and exercise. And part of this is I love this figure. Mostly because I enjoy the history of exercise physiology. And this is a figure from Sid Robinson. So Sid Robinson was a faculty member at IU Bloomington. I walked by his bench every day. He was also a coach he was a track coach at IU. So I walked by his bench every day. So I took a picture of this one day and I tried to incorporate it anytime. Jamais sat in that bench multiple times, I think. But I really like this, and it's also interesting, and I like to make sure and impart on my students, this paper was published in 1952, and it's super relevant to what we do today. So here we have glomerular filtration rate during rest for 40 minutes, and then they walked for another 60 minutes. We have it in different air temperatures. So 25 degrees C, about what we're sitting in now. You can see relatively high GFR, they start to walk, it goes down a little bit. Now, we have people sitting in 50 degrees C air, so that's very hot. That's a lot hotter than what you would experience even in the summer. You see GFR is a little bit lower, right? So they're a little bit hot. You start walking, GFR falls a little more. Now, we're dehydrated and sitting in that 50 degrees C air. You see a reduction in GFR and a huge fall in GFR when you start to walk. So there is a graded response. These things are worsened when they're layered on. In this state, we could have had a very similar figure for kidney blood flow. You see a very large reduction in global blood flow to the kidneys. Now, what's interesting is that at least data in dogs indicate that only the renal cortex sees reductions in renal blood flow, not the renal medulla, which is probably pretty good because the renal medulla doesn't have a lot of blood flow going to it anyways. And also then we have our fluid regulation hormones are up. So vasopressin is elevated, the RAS system is activated, and we also have renal sympathetic nerve activity also goes up. So, so here we have much more sodium being reabsorbed. Okay, so with that background, I'd like to transition to what acute kidney injury is defined as. So AKI is defined as a rapid reduction in kidney function. And clinically, this is when it occurs within 7 days. So imagine someone that's hospitalized in the ICU, you see a reduction in kidney function that's really unexplained. So the clinical criteria are a reduction in GFR and a decrease in urine production. So when we're trying to study acute kidney injury in the context of heat stress and or dehydration and or exercise, we have a problem. Because the normal physiology of these instances actually satisfies the clinical definitions. So how do we address this? So we use acute kidney injury biomarkers or AKI biomarkers. And the idea here is to differentiate kidney function from injury or damage or even ongoing repair. Now, this is a really big area within the nephrology world. And what they're trying to really identify is the renal version of cardiac troponin, right? So can they measure one thing and figure out whether someone is having some sort of pathological event or whether their GFR is falling? So it's actually a huge area of research. We kind of went in and said, I wonder if we can apply this to our work. So that's what we try to do. So here we have a couple of the biomarkers we're going to really talk about today. There's a panel of a bunch of them, but out of simplicity, we're going to talk about largely two. This is urinary NGAL. I'm just going to call them by their names here because they're really long. But we use urinary NGAL because it's the most widely studied AKI biomarker. This is across patients. This is across animal models. This is in healthy people, you name it. And it's a marker of tubular injury, and it largely increases due to the activation of bacteriostasis. And then the other one, which is arguably where we put a lot of focus, not arguably, we have put a lot of focus. It's arguable whether it's the best decision or not. But this is a product of IGF-BP7 and TIM2, and note they're both in the urine here. And these are markers of cell cycle arrest, which is really a protective response during injury. And why we focus on this IGF-BP7 TIM2, which has the trade name of Nephrocheck, is it's the only FDA-approved biomarker for the risk of AKI. So in this event of trying to find this renal version of cardiac troponin, what they tried to do is figure out if you measure this, you may be at higher risk of AKI happening, right? And so when I read all of this, I thought, this is perfect. We're not actually inducing AKI, but what if we measure something that may be relevant, have some sort of clinical relevance for the risk of developing AKI down the line? So that's why we focus on that. There is some clinical utility to the AKI biomarkers outside of the hospital. That's shown here in this figure. There's a number of different papers that have done this now. These are samples from Nicaraguan sugarcane workers where they show that early in the harvest, urinary NGAL levels are relatively low. Late in the harvest, you have a rather marked rise. And these are people at risk of chronic kidney disease of unknown origins. So there does appear to be utility with this. My former PhD student, Chris Chapman, organized some of the AKI biomarkers in this way. And so we're not going to talk a lot about some of these things down here, but hopefully what you can appreciate is with a panel of these markers, we can maybe start to get a picture of not only whether people are at risk, what's the potential location of that injury. Most of the injury is tubular in nature. And then also potentially understand the etiology of this tubular risk. We've even expanded this now to more things that more people associate with things like oxidative stress. So T-bars, TRX1, some other measures where we measure it in the plasma, and we can measure these things in the urine and try to get an idea of what's actually happening in the kidneys. So here I'd like to transition to some of our data where we've used these overall markers of AKI risk and relate them back to people getting hot and dehydrated. So with this, one of the studies that was recently published by a postdoc in our lab, we wanted to test this hypothesis that AKI risk is increased during prolonged exposure to heat stress that's experienced during heat waves now. And then we also wanted to see kind of the idea is trying to have a forward-facing view. We know it's going to get hotter. So why don't we see if this kidney injury risk is going to get worse during future heat waves? So this was actually a retrospective analysis of a paper we had published previously. We had people sit for 8 hours in hot and very humid environments. So 32 degrees Celsius, which is like moderately warm. 35 is pretty warm. We see that all the time in the summer, but very humid, 95% humidity. And these result in wet bulb temperatures. Think of wet bulb temperature as an index, like heat index, right? So what it actually means to the body. This results in wet bulb temperatures of 31 degrees Celsius and 34 degrees Celsius. Now, it turned out that 31 degrees Celsius is actually the current maximal wet bulb temperatures that are experienced during heat waves in certain locations throughout the globe. So these happen today in certain spots throughout the globe. A few of these spots are in the United States. And then this paper came out by Koffel et al. That actually said, by the end of the 21st century, heat waves are going to approach a wet bulb temperature of 34 degrees Celsius, which is wildly hot and wildly humid. And so we wanted to see how this might translate to acute kidney injury risk. So here on the left side, we have our increase in core body temperature. So people are hotter when they're exposed to the 34 degrees C, but they are slightly elevated. Core temperature is slightly elevated when they're exposed to 31 degrees C wet bulb. And they're not allowed to drink during this. So we did see sweating, of course, and reductions in body mass, which is an indicator of total body water. This is nude body mass. So you can see people were about 1.5% dehydrated in the 31 degrees C trial and close to 2% dehydrated in the 34 degrees C trial. So how did this translate to our marker of AKI risk? So here we have our NEPHROCHECK or IGF-BP7 TIM2. On the bottom, we have our 31 degrees Celsius condition, and on the top, we have our 34. We took urine samples before at 4 hours of the heat stress and 8 hours of the heat stress. So what I'd like to note is at 4 hours, this marker of AKI risk was elevated in both trials. It's hard to see it, particularly in the 31 degrees C trial, because I'd like to note that the y-axis is the same in both of these. But hopefully what you can appreciate is that this AKI risk is much higher in the 34 degrees C trial as it was in the 31 degrees C trial. So that's shown over here where you have the mean difference between the 31 and 34. So it does appear that these prolonged exposure to heat wave-like conditions that are evoking heat stress and dehydration is increasing the risk of developing acute kidney injury. Now, we also are currently doing some work where we're bringing this back to more occupational scenarios, where we're looking at this in the context of physical work in the heat. When I say physical work, in the lab, we really just have people walk on a treadmill at certain rates or certain VO2s. But we are starting to do more work that may be more occupationally relevant. We're starting rowing, which is really fun, but subjects don't like it very much. So this is a part of our current R1 through the CDC, where we're really interested in understanding whether the occupational heat stress recommendations protect workers against acute kidney injury or the risk of developing acute kidney injury. So this is one of the studies. So what these recommendations do is they prescribe work rest ratios. So 15 minutes per hour, 30 minutes per hour, 45 minutes per hour, or you can work the entire 60 minutes per hour as a function of how hard these people are exercising. So this is the rate of metabolic heat production. Think of this as absolute VO2 or oxygen uptake, and how hot it is. So this is another index. What bulk globe temperature is another index of the ambient conditions. So there's a lot of data here, but what I'd like to first focus on for this talk is comparing these two conditions. So we employed this is a compliance scenario where they're working for 15 minutes per hour at a given rate of metabolic heat production. And then we also employed a positive control. So in this case, it was much hotter, but they actually did the same amount of work, same exercise intensity and everything in this condition. And so with this, we tested this hypothesis that AKI risk will be lower in the heat stress recommendation compliance scenarios compared to a work match condition. And perhaps based on what I showed you before, it's not too surprising. So sorry, here we have the mean core body temperatures. You can see they got hotter in the non-compliant condition. In this case, they were allowed to drink throughout, which is also compliant with the hydration recommendations. So they didn't lose very much body weight. So they were not very dehydrated, but they were a lot hotter. And perhaps similar to what I just showed you before, it's not surprising that we saw higher AKI risk in the non-compliant, so the hotter condition, compared to the cooler condition. So with this, I think it's fair to say again that even with exercise in the heat, when you layer in exercise, it does appear that heat stress and dehydration contribute to the risk of kidney injury. Okay, so using this as a background, let's transition to some of our work related to how heat stress may increase the risk of developing AKI. All right, so I'll give everyone a quiz on this later, but we're going to walk through it. But this is our overall schematic. I think I talked with some of you all throughout the day. I'm not a renal physiologist. I've never been formally trained in renal physiology. So when I first started doing this, you had like a nice lines showing, okay, maybe this is what's going on. This is our most recent rendition. And so it's really, I found very complicated, but we're slowly getting there. What I'd like to point out is that these processes are happening for a good reason. The idea is we need to conserve water, right? But there may be some negative ramifications, particularly when in very severe instances or when these situations are repeated. But they really center on this instance where we have a supply-demand mismatch. We have relatively low blood flow, particularly to the cortex, and we have an instance in which we're reabsorbing a lot of sodium. Sodium reabsorption via the sodium-potassium pump is a very energetically expensive process. So there have been a number of studies that have looked at various aspects of this. Most of this work has been in preclinical models, but we've started to attack this from the human perspective as well. Today, we're going to talk about a few of the studies that fall within these boxes, and we'll go through these now. We'll start with the relative roles of heat stress and dehydration. And this is one of the studies Chris Chapman did as part of his PhD, where he wanted to test this hypothesis that heat stress and dehydration independently influence the risk of AKI during exercise. So here was his conditions. We had a hot and relatively humid environment. These are conditions that mimicked some of the conditions that the workers in Central America experienced. They walked on a treadmill at 55% of their estimated heart rate max, which is the work intensity that's been observed in these same workers. This is what it looked like. So we do all of our work, most of our work in an environmental chamber. Think of it as a metal insulated box with really good control of temperature and humidity. We have 4 conditions: a control condition, so they receive no cooling or water; a drinking condition where we receive full rehydration; a cooling condition in which they received cooling, but they were not allowed to drink. So underneath this blue tight-fitting shirt, there's a tube-lined suit, which I have pictures of later, that we're perfusing with cold water, like really cold water. And the idea is we're going to help, that'll prevent the rise in core body temperature or attenuate the rise in core body temperature. Then we had a condition in which people drank and were cooled throughout. So this is just a table showing that these conditions worked. They got pretty hot, pretty dehydrated in the control trial. In the cooling and drinking trial, they didn't get very hot and they didn't get dehydrated. And then the cooling and drinking were right in between. So here we have our marker of acute kidney injury risk, our urinary IGF-BP7 TIMP2. In this case, we collected urine at 4 time points: before they go into the environmental chamber, immediately after leaving. So they did 2 hours of walking on a treadmill in this space. Then we had them wait, and we took another urine sample 90 minutes after that. This is our recovery time point. Then we bring everyone back to the lab 24 hours later. Really, this is a safety check to make sure these markers are coming back down. What we don't want is this to stay elevated, because then we might actually be damaging the kidneys. So what was interesting is our original hypothesis is all based on these data, and we saw no statistical differences here. But Chris, after reading more about what these markers are, they're not actually used to look at changes over time. They're spot checks at any given time. So again, very similar to that clinical utility. If you just look at the maximal increase in this IGF-BP7 TIMP2, he's published this in a review he did, you can see there may be something going on here. So there was a greater, the peak increase in this urinary biomarker was higher in the control condition compared to all of the other conditions. One of the other things that these biomarkers allow us to do is try to identify perhaps where the injury is occurring. And so what you can see here is when we split out IGF-BP7 and TIMP2 and we look at it at these same time points, you can see we got a marked rise in the control trial in our IGF-BP7, which is indicative of proximal tubular injury, but there's no such rise in TIMP2, which is indicative of our distal tubular injury. So it appears that most of the injury is in the proximal tubules, and turns out this aligns very well with the animal literature. So just to summarize this section, it looks like AKI risk is dependent on the magnitude of both heat stress and dehydration, and one's not necessarily more important than the other, but you probably need to have both. But what we haven't been able to do just yet is to try to understand the independent effect of exercise. I would suspect that exercise alone would make all of this much worse. Next, I'd like to transition to some of our work on renal blood flow. And we've done a number of studies in this area, and our overarching hypothesis is that heat stress modifies renal vascular control. So the kidneys are somewhat challenging to measure the renal vascular control of them, especially relatively non-invasively. So we've employed renal Doppler ultrasound to do this. So here we have an example of what the right kidney might look like when Cincinnati. This is very deep, so you can really only see branching and velocities when you're in the color mode. But here we have the right renal artery, and then it branches out into the segmental arteries. And this allows us to measure renal blood velocity. Chris Chapman has done a lot of analyses to show it was remarkably more reliable than I ever thought it was going to be, both within day, between day, you name it. So it does seem to at least be reliable. The validity of it, we don't super know. So I will say that caveat across the board. So one of the things that we were interested in is whether heat stress modifies the ability of the renal vasculature to vasoconstrict. So one of the first things we did is we had people do a cold pressor test where we put their hand in agitated ice water. This elicits largely a pain response. You get a huge increase in sympathetic nerve activity. And what you should see is vasoconstriction occurring in the renal artery. And so sure enough, when we measure it with Doppler ultrasound, we see rather large increases in renal vascular resistance. So this is our mean arterial pressure divided by the blood velocity. So it's really an index of vascular resistance. So from this, we've really utilized the cold pressure test as a way to test the renal vascular responsiveness to sympathetic stimulation. So one of the studies that Chris wanted to do was to investigate the effects of heat stress on this response. So we did this by using water perfused suits where we heated people up. So these are tube-lined suits. We connect them to pumps and we pump hot water into them. So we have a student modeling that for us. And so in Chris's study, we measured renal blood velocity during the cold pressure test at baseline and then when they were hot. And this is pretty uncomfortable. Let's see. Okay, so we measure this in the right renal artery and also in the segmental arteries. And so here we have we do the cold pressure test for 2 minutes, and we have the increase in renal vascular resistance on the y-axis. And hopefully, what you can see here is that during normothermia, so at baseline, you get a rather robust increase in renal vascular resistance, whereas during hyperthermia or when you're hot, that increase is attenuated. And that's true whether you're in the renal artery or the segmental artery. In fact, it looks like the responsiveness might be a little bit more exaggerated, or the blunting might be a little more exaggerated when you're in the segmental artery. More recently, we've given protein, so oral protein, just whey protein, which induces renal vasodilation by inhibiting tubular glomerular feedback, which is again my renal physiology is getting better, but again, it still seems really complicated to me. And so in this instance, we wanted to look at whether heat stress or normothermia, whether heat stress modified the renal vasodilatory response. So here we have the segmental artery response. You can see a reduction in vascular resistance, but it did not differ between heat stress and normothermia. And that's also true in the renal artery as well. So to summarize, looks like heat stress attenuates sympathetically mediated vasoconstriction in both the renal and the segmental arteries, but does not modify the vasodilator responses. And then we still have a number of questions here that we're not quite sure. Mostly, exactly how does this align to AKI risk? We're really early in trying to understand how heat stress modifies the regulation of blood flow in the kidneys. Finally, the last little section, which I think are just kind of fun studies, so I wanted to bring them up, relates to how this pathway, this fructokinase pathway, may contribute to acute kidney injury risk. So here, this pathway really starts with hyperosmolality in the proximal tubules, where you see the enzyme aldose reductase is upregulated, and it really triggers this pathway where you can get endogenous production of fructose. And the idea that this may be involved with heat stress really comes from the same group where they had a fructokinase knockout mouse. They exposed them to this 7 hours per day, 5 days per week for 5 weeks. And they compared the responses to a wild type mouse. You could see in both instances, they were not allowed to drink during the heat stress. So they both always lost a lot of body water. They got the same amount hot. But the fructokinase knockout mouse had better kidney function, and the fructokinase knockout largely prevented proximal tubular injury. So based on these data, this group followed up, and they said, well, what if we give rats, I think there was some rats, but I think that's a picture of a mouse. But what if we give rats a soft drink-like beverage and expose them to this repeated heat stress over 4 weeks? What happens? And the idea being you add substrate to that pathway. So you add fructose, exogenous fructose. And sure enough, there's a number of different figures here, but this is just one of them. We see N-gal protein expression in the renal tubules is much higher whenever you've given these rats a soft drink like beverage compared to a taste control. So this is actually the study that got us into this whole mess, because I read the animal in rats and I was like, man, I wonder if that happens in humans. So after a bunch of pilot testing where we drank a lot of Mountain Dew hanging out in the heat chamber, we actually decided to go ahead and run a study. And so this was Chris Chapman's first PhD study, where he wanted to test this hypothesis that drinking a soft drink, so this high fructose corn syrup sweetened beverage, we were using a big hammer, so we also used a caffeinated beverage, that it elevates, during and following exercise in the heat, elevates markers of AKI. This was our model. We measured urine pre. We had people work in the heat for 4 hours. We measured urine post, and then we also measured 24 hours post. So the work looked something like this. It was a little more occupationally relevant. They walked on the treadmill for 30 minutes. They did a lifting task, a dexterity task, this hammer task, which was a lot of fun. And then we rested for 15 minutes. And during that rest, they drank 500 milliliters of either water or Mountain Dew. Okay, so yes, they drank 2 liters of Mountain Dew throughout that period of time. And by they, I mean, I was the first subject for this. And I will say, drinking Mountain Dew was way better than drinking water in that case, because you're so hot and tired, and it just tasted good. But we chose Mountain Dew because it's got the most high fructose corn syrup of any soft drink, okay? It's also lime green, therefore it cannot be good for you. So what did we find? This is just how hot people got. So they got the same amount hot, they got the same amount dehydrated, which wasn't very dehydrated. So they drank the same volume of fluid. Here we have our marker of kidney function. So we had data before, immediately after, and then 24 hours post. And you can see that this is in the change in serum creatinine. And when you're drinking soda or the Mountain Dew, you have a greater reduction in kidney function. But what was interesting is that when we measured urinary NGAL, we saw no difference. Now, one thing I'd like to note is that you notice in our other work, we have this recovery time point. And the reason we have that recovery time point is we collected all the urine between post and 24 hours. And so when we saw this result, we were kind of like, oh, okay, well, maybe we're wrong, right? That's okay. But we have all this 24-hour urine. Maybe we should measure urinary NGAL in that mixed sample. So it's a large volume of fluid. And sure enough, we get a small but statistically significant elevation in this overnight urinary NGAL. So it's very diluted because it's a huge volume, but it was, I wish I had the individual's values up here because it happened in everyone. So this led us in the subsequent studies to build in this recovery time point, which is really where we see all the really exciting stuff happen. So we went and did a quick follow-up study where we wanted to see why this might be occurring. One of the hypotheses was that when we activate this fructokinase pathway, it could induce renal vasoconstriction. So Chris got really good at the renal Doppler ultrasound. So we gave people Mountain Dew, we gave them water, we measured renal blood velocity, and sure enough, blood velocity falls and vascular resistance is elevated. So it looks like the Mountain Dew does induce some amount of renal vasoconstriction. When we layer on the cold pressor test, so here we have our 2-minute cold pressor test, we see at least in the first minute, we see a heightened increase in renal vasoconstriction. So it does appear that the vascular tone seems to be shifting more towards constriction when you have this huge amount of high fructose corn syrup. So hopefully I've convinced you that consuming a soft drink during and following exercise in the heat can increase the risk of developing AKI. This risk is mediated likely by this fructokinase pathway by adding exogenous fructose as substrate, and which probably promotes renal vasoconstriction. It's also possible, animal data and some of our data indicate that vasopressin and uric acid may also be involved. But I should note that there's no evidence in humans or even animals that this pathway is, except for the knockout mouse, that this pathway contributes to AKI risk in the absence of giving a substrate. So we still don't know whether that's the case. We're starting to look at some of this in our animal work. Okay, so just to summarize, hopefully today you can walk away and say heat stress, dehydration, and maybe even with exercise on top of it increases the risk of developing AKI. This is probably renal tubular injuries, and it's likely in the proximal tubules. This AKI risk is dependent on the magnitude of dependent on the combined magnitudes of both hyperthermia and dehydration. So how hot you get and how dehydrated, and one's no more important than the other. There's a bunch of mechanisms. Remember, we'll give you a quiz over all of the schematic later. I showed you today that it looks like heat stress modifies renal vascular control. There may be some involvement of the polyol fructokinase pathway. I didn't show you data here, but there's some evidence that hyperuricemia, oxidative stress, inflammation are all playing a role here. And most of our work really is centered on understanding these mechanisms and modulators is important because hopefully we can develop countermeasures, which is what we're trying to transition to now. So with that, I'd like to say thank you. Like I said, I really have enjoyed my time here. Looking forward to going to dinner tonight. And if anyone, students, faculty, whoever wants to get a hold of me, there's my email. And the lab made our lab join Twitter. So please follow us if you can. So great. Thank you very much.
So in a lot of your work, perhaps it was mostly men. Can you talk about any potential sex differences that you would expect to see? And then also when you're using your soda, can you comment on the role that caffeine may be playing versus if you were to use diet soda?
Yeah. So aside from one study, which was the retrospective 8 hours of heat stress, that was all men. That was a kind of offshoot of a study that was funded by the DoD, and the Navy wanted men because there's like one woman diver in that group, which we tried to argue actually for putting women in, and they said no. But all of them are half men, half women. Now, what's interesting is we're very interested in whether there are potential sex differences. We actually have the data. We just finished analyzing them now. And so hopefully we'll know, like they were analyzed yesterday. So hopefully we'll know at the next APS meeting. That's our plan. But it looks like from the epidemiological data that there are sex differences. But sometimes, for the most part, men are at higher risk. But there's actually 2 papers that show women are at higher risk. And there's a number of reasons for that. At least with ischemia, reperfusion injury in the kidneys, testosterone is bad. So that's part of the men's side. But the women's side, there's just less renal functional mass. And so it may be that a given reduction in blood flow may manifest worse. So regarding caffeine, absolutely. We actually did a study as part of the blood velocity, the renal blood flow stuff. We did a study where we used caffeine-free Mountain Dew and did not see. Sorry, what did we see? We saw. I'm trying to remember. We were able to rule out the role of caffeine. It looks like it's mostly high fructose corn syrup. So we had a condition where it was like we have water, regular Mountain Dew, throwback Mountain Dew, which is sweetened with sucrose, not high fructose corn syrup, a diet Mountain Dew, and then a caffeine-free Mountain Dew. And everything was happening in the high fructose corn syrup one. So, yeah.
So I'm thinking about from the perspective of like elite athletes that end up getting acute kidney acute kidney injuries are bound to accompany that. And the first thing you do too is just throw them into an ice bath. You have to pull down, really want to pull down brain. But is that necessarily healthy for the kidney? Want blood flow to reduce the kidneys, or would you want like that?
Yeah, so with heat stroke, in that case, so the treatment for heat stroke is you dump them in an ice bath. What's interesting is that heat stroke causes AKI as well. So treating the heat stroke, getting body temperature lowered is by far the number one thing. If you do that, then they don't get acute kidney injury. We have measured So part of the renal blood velocity stuff, we measured it when we heated them up and then we cooled them back down, like aggressively cooled them back down, and all completely normalized. So it wasn't any worse. It wasn't worsened by the fact that we cooled them. So I think the main thing is cool them. And at some point, that's the acute kidney injury is probably the least of their worries if their central nervous system's frying.
Couple of questions. Very nice talk, Doug. To follow up on Mark's comment, have you considered measuring those markers of acute kidney injury risk? Individuals who train for endurance events in the summer.
Yeah.
Do they experience those increases?
I'm not sure about training, but I know people have done it like across a marathon, across an ultra marathon and stuff like that, and they do spike. And so there's this debate as to whether it's bad, right? My argument is it's not, because being aerobically fit and physically active reduces the risk of kidney disease. So it's, again, I think it is actually analogous to like cardiac troponins. Like you measure cardiac troponins after that race, yeah, the heart's been stressed, but it's not a problem.
Each training session, which usually each training session is associated with at least a 2% loss in body weight associated with dehydration in the summer.
Maybe, yeah.
That could also create
it could, yeah. I think the difference between what the athletes are doing and what workers are doing, let's say, is so different. Workers are out there 8 hours a day, 7 hours a day on a short end, and they're working really hard. They're working at the average is 55% of their estimated heart rate max. Like they're occasionally at 80, 85%, right? And they're doing this day after day, week after week. And even if you're training for a marathon or something like that, that magnitude is still half of what the workers are getting. And then when you run your race, you're going to take some time off, right? When the workers aren't doing that.
I have a follow-up question: is have you considered measuring individuals that have their sweat inefficient? Sweat efficiency is difficult to measure, but
Yeah.
those that are sweat inefficient, those are individuals who sweat in excess relative to how much they evaporate.
Like hyperhidrosis.
Yes. Are those at greater risk? Are they at greater risk for kidney?
I mean, in theory, right? If they're not able to drink, they're going to lose more body water. So in that case, they may be at higher risk. But the question is how many, those people, that may be an interesting case study. I'm not sure there's a ton of people out there that are hyperhidrotic that are doing those things. But anything that's going to make someone hotter or more dehydrated, I think could, in theory, put them at greater risk of AKI.
Nice. I'm interested in the variability. The first study you showed, the 31 versus 34, it plays out. Everybody's going up. Is that differences in age? Is there body composition difference? What predicts?
So in those figures, these were not normalized to. So when you measure in urine, it's quite challenging because you get huge differences in urine concentration and urine output. The standard practice is to normalize to urine flow rate, or essentially urine production. So everything we do is normalized to that, and that really reduces the variability. In this case, when we're presenting, the FDA approved marker, there's no normalization involved, and so that's why there's that variability. We then we have a bunch of different markers, and we're looking at pathways, and if you look at that measure when it's normalized, the variability is much smaller. At this point, we haven't ventured into really like heterogeneous groups and stuff. We're really trying to just figure out what's going on. We spent a lot of time trying to figure out what to measure, but I think now I'd like to think we figured that out, and now we're going to move forward.
There's also a co-transporter for fructose and uric acid.
I feel like I've read it, but I'm not. Yeah, that'd be interesting.
On low fructose diets, even if you don't increase intake, uric acid goes up.
Yeah.
You're not losing fructose through the urine.
Yeah.
Uric acid goes with it.
Yeah, that's interesting.
So the idea that you push that system with the fructose.
Yeah, and that's an area hopefully we can do more of. But I was shocked to find out like the kidneys metabolize like almost 10% of the fructose, right? So you think of it's all in the liver, but it's not. So, and so that the fact that that pathway exists and is beneficial actually against hyperosmolality, but when it's chronically active, it looks like it's bad. So hopefully that if you could send me that, it'd be really interesting. Yeah,
It has that.
yeah,
to be able-
absolutely, yeah. That's good there.
So in your soda stream, match the sodium content in the water to that soda. If not, do you think that that salt has any impact on your?
Yeah, we didn't. I don't. There's not a lot of salt in it. There's a little bit. You're right. I don't think it matters. We haven't done it in the context of like the AKI biomarkers, but when we go back to our the renal blood velocity stuff, when we give that throwback Mountain Dew and we gave the actual Mountain Dew, all the effects were in the actual Mountain Dew. And the only difference between those, same calories, same everything. The only difference is high fructose corn syrup versus sucrose. So, yeah. I probably should have, I forgot, I probably should have, I should remember there are nutrition people here. And when I give the soft drink stuff, it's like, oh, yeah. I was like, but man, I should have made sure and brush up on that. Go ahead. Yeah.
So how would you expect those like integrated like a sauna like that? I mean, I guess you don't drink whatever that also produces.
Yeah, I think it depends. It's a function of time and how hot you get, not necessarily the ambient conditions. And so, if you're in a super hot sauna, most of the time you get out before you really get that uncomfortable. And so you're probably fine. Now, if you're trapped in there for a long time, then you have a problem. So, but it's a good point, especially with like heat therapy and that kind of really being a hot topic. That I think that's important in that context, but it doesn't look like it's doing anything bad necessarily. Thank you.
So have you seen any evidence that like stress or biomarkers there or balance in cells or podocytes?
I don't think I have not seen anything. Yeah, I don't think so. Not in the context of the kidneys. I'm trying to think of outside of that. But I don't think so. I would say there's not a lot of work. Only one group is really doing heat stress kidney work and work in aging. So yeah, I don't think so. Why do you ask?
The glycolysis is important for vascular function.
Right. Yeah, right. Yeah, I don't. The reduction in GFR, I think, is physiologically mediated. It's not like an impairment occurring, but what's could be happening is like at a local level. So I think if you think of it as our hypothesis is that there's the whole kidney is not hypoxemic and depleted, but there's like pockets where there's supply demand mismatches going on, and maybe in those instances, I would suspect, and I was having a conversation with my renal physiology collaborators that there's likely kind of profuse endothelial dysfunction and disruption occurring in those specific little areas. We don't know yet. Oh yeah, great. She's collecting data. That's awesome. Yeah. Yeah.
So, based on question, there tends to be an ethnic difference in kidney functions, but do you have that data?
Yeah, we don't. So I forget who I was talking with someone. We really one of the key challenges, I think we were talking about this, one of our key challenges in Bloomington, Indiana, is we have a severe lack of diversity in our student body as well as in our general population. So we're working in that area to try to address some of those questions, but we don't have the data. I would suspect, we've had a lot of conversations, there's a lot of like differences in urine concentrating ability and, you know, RAS systems and stuff like that. I would suspect that, and the risk overall of AKI and chronic kidney disease is much higher in certain ethnic groups. So I would suspect there would be something there, but we don't have.
Think about all the folks, stretch guys in Texas, and then they're all working, yeah, 6
Yeah.
hours a day.
Yeah.
They all drinking soda.
I know.
A lot of them do. I mean,
Yes.
you think it's crazy. My brother-in-law is an electrician, and so he spends a bunch of time in attics in the heat in Kentucky, right? And he had his apprentice, and you know, he goes in, gets water, and you know, maybe a Gatorade, and his apprentice comes out with 2 big gulps, and he's like, "What you got there?" He's like, "It's Big Red," right? Like, if anything is worse than Mountain Dew, it's got to be Big Red, right? Like, I think it looks like neon, and so yeah, he just chugged those. He was like, it was 120 degrees in the attic, and he's just up there just chugging down Big Red. So I think that there's a lot. The problem in the US is the reporting. A lot of the people that are at the highest risk are migrant workers and undocumented workers. And so it's hard to understand what's happening. There is some work going on, particularly in the Southeast, like in agriculture, as well as in California, where they're actually getting into those situations. And the other thing that we think of is heat stress is like, oh man, I got hot for a while. Then you can go inside. In these cases, people are working in really hot conditions and they go back to a shack that has a fan. You know, they're not getting a break. And that's something that it's hard for us to simulate in the lab. We're designing a study to do that. So hopefully, you know, hopefully we can get volunteers to sleep in the lab, but we'll see how it goes.
So you commented that you have CDC funding, and you have that graph that show kind of the A, B, C, D. So are those guidelines currently from the CDC that says at a certain temperature you should really only work for 60 minutes?
Yeah, they are. So it's NIOSH, National Institute of Occupational Safety and Health. So it's an institute that sits within the CDC, and it's kind of a weird institute. It's like a little bit research and a lot like training and stuff like that. So but it's distinct from OSHA, if that makes sense. So OSHA makes the rules, NIOSH gives recommendations. So the recommendations, which are based on science, and our data indicate that like they work. People don't get super hot. They don't get super dehydrated. These are out there, but they're not enforceable. OSHA does not have a law or a regulation to hold employers accountable for heat. Unlike, so, you know, there may be like air exposures, asbestos or whatever, like those are rules. And if the employer breaks that rule, there are consequences. There's no such thing for heat stress. Now, if people were to abide by these recommendations, we would get no work done ever. So the issue is they're too conservative probably, and we need to think of more innovative solutions than just saying, okay, you can't work a certain amount of time. That's probably why there is no rule, is because employers and probably OSHA knows that if they enforce them, you're not going to get any work done. So, but yes.
So your role is to identify if at those stages it causes injury and then maybe to develop a countermeasure.
Right. So we wanted to first understand the overall premise of the grant is there are rules for heat stress, which is the biggest part, this prescription of work rest ratios and stuff. There are recommendations for hydration, and which is both volume as well as what they drink. So we're finishing the first 2 aims now, and then we're just in the last year. So we're working on the 3rd one. And so that's kind of where we are. It looks like actually the hydration recommendations give you too much fluid, like people can't drink that much, so we've had to scale that back. So drinking ad lib is fine. And then the last part we want to understand, the recommendations say if it works over 2 hours, you have to drink something containing electrolytes. But what we're really interested in is Powerade is sweetened with high fructose corn syrup, and Gatorade is glucose. So we're going to try to understand that and some other constituents. But we try not to, like, I appreciate sometimes we're like hydration toes the line of nutrition. And so we try not to be too nutrition-y. Then I get in way over my head. So, but we do have a nutrition person on the grant, so that helps.
One more quick question. So in one of your studies, you were giving patients protein, increasing oncotic drive, kidney physiology.
There you go. Yeah.
Would that help? Would that serve any kind of protective role? If you would add some protein?
Yeah, that's a really good question. So this idea, so the protein, the bigger part of that project was this idea of renal functional reserve. So what if you give protein, GFR goes up, and with a bunch of mechanisms, one of which is oncotic drive. And so there's some people that think or have proposed that if you infuse amino acids when someone has acute kidney injury in the hospital, let's say, you can help them tap into their reserve and preserve kidney function. So from at least the kidney perspective, I just had a conversation with my PhD student who wanted to give protein immediately after exercising the heat and see if what happens to the acute kidney injury markers. We haven't done the acute kidney injury markers yet because that was all passive heat stress. That was like one of our COVID babies. So we couldn't get in the environmental chamber. So we heated people up with suits. So I suspect that may be protective, at least against the drop in GFR. How that translates to other markers, I'm not quite sure yet. But it may be beneficial because you may be reducing the energy demands of the kidneys with the sodium stuff. The other thing that may be beneficial, which we don't fully know, I think we talked about this a little bit before, but if you add amino acids to a beverage, it will aid the transport of fluid through the intestine into the body. Now, none of those data have been done during exercise, heat stress, dehydration, any of those states which have reduced gut perfusion. So whether that holds in that state, I'm not sure. But certain companies would have you believe that's the case. Yeah. Thank you.